Thank you for that introduction. I wanted to start by just saying that what we're presenting you with today is a kind of study, and this study comes out of an exchange between myself and Tamar. And I see our experimental collaboration as a process, and this process will have multiple different outputs, which each take different forms. And so this lecture is just one form of output that we're generating. Tamar and I are also co-authoring an academic journal article around this research. And then the main output is a feature, a larger, more ambitious film project, which I'm currently in production with. And I just wanted to say up front that in a way, we're thinking about all of these elements as a research experiment, which is inspired by the Freemartin, but also by how other people have engaged with the Freemartin as a tool of their own research. So, let's start with an image conjured by writer and activist Sophie Lewis in her introduction to amniotechnics, a chapter from her book Full Surrogacy Now, which she dedicates to water. Lewis describes the art of the deep water diver, slowing their heart rate in preparation to swim by imagining an in utero state. For the deep water diver, manifesting such a state is a way to calm themselves to perform their task. And this conjuring of a time before fear serves a very practical function of producing in the diver a kind of non-antagonism towards water. Like Sophie Lewis, what interests us are the many different kinds of people that have likewise been preoccupied with the amniotic state, and the attempts that have been made to analyze the kind of in utero manufacture taking place within amniotic fluid. How are bodies brought to life in water? And how has knowledge in this field involved the senses? How have taste, touch, sound, and smell been ways of coming to terms with this deep manufacture? Much of this study takes place in the early 20th century, as scientists in Europe and the US tried to test the limits of human growth and reproduction, and during the race to define the endocrine system, what we know today to be a system of glands that produce and secrete hormones within the body. Around the same time, the human body was gradually being separated from its own waste, including feces and urine. Houses were increasingly fitted with flush toilets, enabling excrement to be removed from the home, and with it, the idea of making bodily waste invisible became an expectation. Flush toilets became symbols of modernity, and in this context, scientists searched to find value in urine, as it was found to contain valuable sex hormones that could be used for enhancing fertility. Pregnancy urine, for example, of either women or horses became a key resource for extracting hormones. And as such, relations were established between pregnant women, the pharmaceutical industry, and patients of fertility clinics all over the world, prompting a flow of biological materials between the home, the farm, and the laboratory, as bodily waste became the basis for the development of fertility drugs. Human and animal urine became targets of collection campaigns and leading resource material for the booming pharmaceutical industry. Such movement and attention to the waste of the body is indicative of the period. But our plan here is to embrace a slightly different narrative of waste. The protagonist of this story of human endeavor is non-human. And the real site of conflict is the question of how the animal is born intersex, which for hundreds of years has perplexed many. Here is an image of our elusive protagonist, the Freemartin. Born almost always twinned with a male bull, the Freemartin's sex has been referred to as mosaic-like, as the animal will likely possess external female genitalia and internal male gonads. This is as a result of early exposure within the placenta to the male hormones of its twin. which influences the development of the Freemartin's female reproductive organs. Most have an almost unrecognizable udder and small teats, and the Freemartin is often born sterile. For centuries, the Freemartin phenomenon presented the farmers with a problem because, set against the backdrop of industrialization, the Freemartin's infertility defies the logic of the farm. And as such, farmers collaborated on ways to identify Freemartinism as early as possible in order to avoid unnecessary investment in its feed, health, and growth. Elsewhere, many historical actors, such as scientists and sociologists, have turned their attentions to coming to terms with the indeterminacy of the Freemartin's sex characteristics, ultimately in an attempt to extract value from the animal's otherwise waste body. Here we will explore how many significant realizations about sexual difference, endocrinology, embryology, and biological autonomy have been made through experimentation with the body of the Freemartin. And later, how the Freemartin problem, as it came to be understood, was superimposed onto the sociological realm, where attending to the gender trouble of the Freemartin became a tool for reinforcing normative stereotypes and gendered ideas about sex and marriage in the context of the home. The phenomenon of the Freemartin was long known to farmers. As one mid-19th century physician described, in external appearance and form of body, they usually resemble the ox and spayed heifer, more than either the entire male or the entire female of the species. Likewise, Freemartinism in the context of the contemporary farm is still identified using the cow's exterior appearance and behavior. This is apparent in the contemporary transnational farm-to-farm chat room online, where people share their knowledge and tricks for controlling animal reproduction. As one anonymous user on farmforum.co.uk writes, the vulva on a Freemartin usually looks deformed and they often pee to the side or straight up. The same anonymous user adds later down the chat thread, they also sometimes have a slack pouch of skin or a tuft of hair somewhere between the vulva and where the scrotum would normally be on a male calf. We were taught to use a Bic pen to test for Freemartin heifer calves by one of our vets, another user adds. Normal heifers have vaginas that are approximately 30 centimeters long, but vaginal lengths in adult Freemartins are around 8 to 10, making the pen the ideal length for the job. If it goes in full depth, he concluded, we keep the calf. Early detection methods such as the pen test involve deep knowledge of the animal from the inside out. We found parallels between this agricultural method of detecting Freemartinism and the early 20th century drawings by American zoologist Frank Rotre-Lilly, who, like many others, used a form of sense knowledge to come to terms with how the Freemartin comes to be born intersex. We use the term sense knowledge to describe attempts at grasping nature that rely on sensorial cues. Defining the quality of the animal's voice, for example, the taste of its meat, the smell and shape of its body, had for hundreds of years been a way of knowing it. And its sensorial presence was commonly examined through sound, taste, smell, and color. In the 1770s, for example, a Freemartin was killed in Berkshire, England, for the purpose of a sensory experiment. The Luckley community congregated to perform a taste test in order to know if the salvaged Freemartin meat was better than bull. A witness noted that every neighbor bespoke a piece, which turned out nearly as bad as bull beef, at least worse than that of a cow. Elsewhere, the British surgeon John Hunter attempted to characterize the quality of the Freemartin's voice. In his 1779 account of the Freemartin, he noted, it was familiar to that of an ox, which is not at all like that of a bull. It's more of a cow, although not exactly that. Sense knowledge was also employed to search for Freemartinism in other species beyond cattle. The British naturalist and goat expert C.J. Davis, for example, argued in 1913 that intersexuality is common in certain breeds of goats. The main indicator of Freemartinism in goats was its smell. As many goat breeders reported, Freemartin goats developed the characteristic smell of the male and at times they noticed their smell was even worse. And finally, colour became an important tool for trying to understand how and why the Freemartin was born intersex, as can be seen in the Freemartin research of zoologist Lilly, who used colour in studying embryos. Colour, as we will explore more deeply, was gradually established as an important tool in Freemartin research across different fields of knowledge. Case 44, for example, was a 7-week-old Freemartin embryo that arrived at Lilly's laboratory from his experimental Buffalo Creek farm in 1914. The Freemartin embryo was still wrapped in its uterine membrane, and Lilly needed a method for sensing the nature of the Freemartin's connection with its twin. So he began using colour. He injected blue ink into each of the twins' umbilical cords, yellow into the other, and followed the paths of blood circulation. And through an evolving flow of greens, the Freemartin gradually revealed astonishing levels of admixture. Lilly had created a visual map, which exemplified the vascular connections between the male and the Freemartin twin. And with this injection dyeing technique, he was able to argue that materials such as hormones and blood were carried through this web of connections. This sensory aid allowed Lilly to demonstrate that this connection is what makes the female sterile. By tracing the movement of hormones using colour, Lilly set in motion the idea that sex differentiation is not stable and predetermined by what we consider today to be genetic material, but instead, the effect the male twins' hormones had on the Freemartin's sexual development exemplified sex differentiation as a process affected by environmental factors within the womb, a theory that challenged commonly held beliefs of the time. Lilly's account of the Freemartin became his claim to fame. As one of the founders of experimental embryology, Lilly gathered this sense knowledge during the height of the eugenics movement. And it's clear that his preoccupation with the gender trouble of the Freemartin is not unrelated to his eugenics activism, as he went on to use his findings to make claims about sex characteristics in mammals at large. Lilly was more than familiar with the notion of weeding out undesirable hereditary traits, and perhaps even instrumental to the movement of eugenics, serving on the Advisory Council of the Eugenics Committee of the United States, and including a paper on sex hormones, which he presented at the Second International Eugenics Congress in 1921. At the end of his seminal 1917 study of the Freemartin, Lilly explained that the study of cattle is valuable because of the potential implications to human society. The path to controlling human reproduction and sexuality is paved through the work of scientists, he said, and added that the most important task of biology was to determine the means, limits, and subsequent results of such control. It's also perhaps no surprise that earlier researchers studying the Freemartin had already attempted the transposition of the Freemartin problem onto human social relations as a means of reinforcing gendered stereotypes, or more specifically, marriage as fundamentally tied to a woman's ability to produce an abundance of offspring. In the mid-19th century, sociologist James Y. Simpson used hospital records of twin births and marriage registers between 1820 and 1930 to test the success rate of marriages in women who had been born co-twin to males and whether such marriages were productive. He ultimately showed that such women are unproductive in the proportion of 1 in 10, and as such do not exceed the unproductiveness of marriages elsewhere in the community. But despite the failure of the sociologist's attempt to use the sexual incompetence of the Freemartin to reinforce normative ideas about sex and marriage, the tendency to equate human infertility with the category of the Freemartin has continued throughout the 19th and 20th century, as did the need for sense knowledge to grasp the Freemartin's liminality. This can be seen in the revival of Lilly's early 20th century placental dyeing technique, known today as the Solomon technique, which is now used in the context of human twin births. Ink has been replaced by color-coded gelatin dye mixtures, or even lasers. And this method continues to be the most precise way to classify the various complications in twin pregnancy, in which babies are dependent on a single shared placenta. Lily's Freemartin studies have also directly influenced what's come to be known as microchimerism, or the presence of two genetically distinct cell populations in the same individual. This has opened up huge fields of interest into the ways in which fetal cells influence maternal biology during pregnancy, an idea that led to the important realization that a pregnant woman may harbor cells that from the person that gave birth to her, as well as the fetus that she's carrying. This placental traffic, termed feto-maternal microchimerism, is now recognized as a phenomenon which occurs during pregnancy, as well as shortly after giving birth for most women. The placental exchange of biological material across the placental membrane is recognized to have lifelong consequences, linked, for example, to certain types of autoimmune diseases. As such, Freemartin research blows open the idea of biological parenting as a process of segregated authorship. And this research has also been used to destabilize the ethical, medical, legal, and genetic elements of surrogacy, where a woman agrees to gestate on behalf of another person. In the context of Freemartin research, the surrogate carrier can be understood as having a biological connection to the fetus and vice versa, which breaks down any presumed autonomy between the gestational surrogate, the child, and the genetic donor. Hence, Freemartin research has the capacity to bring to light a particular view of gestational surrogacy and kin-making at large, in that the carrier and the person who donates the genetic material could be described as engaging in a polymaternal matrix of collective labor, perhaps forcing a redress of the collective needs and positions of all involved in the surrogacy contract. And perhaps once again, reintroducing the problem of the Freemartin as a form of productive confusion. In addition to using the Freemartin to know and control social norms in these turn of the century contexts, a more contemporary wave of Freemartin research has focused on using the animal as a technology for managing reproduction. Here, the scientific preoccupation with the Freemartin's indeterminate sex characteristics has opened up new possibilities, and many have focused on transforming the Freemartin's waste body into a valuable asset in a process that is known on the farm as heat detection. So, how do you detect heat? When a cow is in heat, its legs will lock in position, and by arching its back, it supposedly indicates a willingness for being mounted, in which case it will stand motionless under the weight of the mounter without pulling away, which is a sign that the cow is in heat. Farmers have learned that ovulation occurs 25 to 32 hours after this encounter, and for centuries, this standing behavior was the only reliable visual cue they had to determine time of ovulation. The animals that are trained to act as heat detectors are commonly referred to as gomers. Gomer bulls give farmers a physical indication of the ultimate time for artificial insemination, and are generally surgically altered through vasectomy to make them sterile. Vasectomy retains testosterone production, which farmers equate with the desire for bulls to mount fertile females. So the farmer uses the Gomer bull to create a visual indication of which cows need inseminating. But surgically altering bulls is expensive and dangerous, and so agricultural scientists turned their attentions to the Freemartin. If the sterile animal could perform as a Gomer, they could replace the bull, and it wouldn't require surgical alteration. And so in the early 70s onwards from across America and Europe, scientists and farmers collaborated to explore whether the Freemartin's naturally higher levels of testosterone would need supplementing in order for the animal to exhibit the so-called desire to mount cows. One such study conducted at the Animal Research Centre in Ontario, Canada in the mid-1980s attempted to offer a scale of heat detection capacity, comparing between Freemartin Gomers, regular heifers, and humans that act as heat detectors. Hormonally treated Freemartins were fitted with chin ball markers, which consisted of a stainless steel spring-loaded ball filled with brightly coloured paint and attached to a leather harness that fixes under the heat detector's chin. Echoing Lily's early use of colour, the successful goma was expected to smear, drag, or splat paint across the heifer's backside as it mounts the fertile cow, providing an indication that the fertile cow is ready for insemination. Fitted with harnesses, the Freemartins were transformed into cow-sized ballpoint pens, and the study recorded the paint marks on their backs. These markings, like expressionist paintings, were designed to provide a gestural representation of the heifer's sexual receptivity. Desiring energies are denied for the artificially inseminated lactating cow, for whom sex and pleasure has been removed. Mounting and foreplay is simply a visual indication that she's ready for insemination. But strikingly, for the Freemartin goma, sex is encouraged. The Freemartin is even redesigned for the job by way of hormonal supplements. Farmers took a gamble on the Freemartin's disobedience and invested in the Freemartin's sexual desire for non-procreative, non-normative sex. But in this particular study at the Animal Research Center, it was found that Freemartins treated with testosterone can detect estrus in cows, but were not actually as effective as hormonally treated heifers. As it turned out, non-androgenized cows were less selective with their mounting activity. And instead, untreated Freemartins displayed more mounting than their active testosterone-treated counterparts, demonstrating that testosterone was not related to the proportion of sexual activity. And once again, the Freemartin confounded expectations. Yet, interestingly, many farmers and scientists continued to invest and collaborate on ways of transforming the Freemartin into a heat-detecting goma. And chin ball markers filled with paint and DIY experimentation with hormonally treated Freemartins remains a major topic on the experimental research in the informal context of contemporary online farming forums, despite the fact that Freemartins seem to make pretty unreliable gomas. In trying to answer the puzzle of why this kind of sense-based experimentation has persisted, we came across a number of recent English and American studies which explain how Freemartinism is a rising phenomenon as a direct result of the industry's insistence on increasing cows' milk yield. Cows which are consistently pregnant during high periods of lactation have a higher chance of producing twins, as milk production produces a hormone which interferes with egg development and increases chances of twinning. We've started to understand this more recent wave of sense knowledge and creative experimentation with transforming Freemartins into gomas as a means of salvaging economic losses from a problem created by industrialization. Recent digital tools rank dairy cows on their genetic merit. And against this system, and devoid of procreative value, the Freemartin can really only exist as a tool to enhance the value of others, as a heat detector. Some Freemartins work around the clock, catching otherwise missed heats, and the animal's temporal pleasure seeking helps organize cattle reproductive cycles in time with milk and meat markets. This type of relationship between time and body is what literary theorist Elizabeth Freeman defines as chrononormative, in that time is used to reorganize individual bodies towards maximum productivity. Chrononormativity, according to Freeman, involves a retemporalization of bodies so that industrial forces come to seem like somatic bodily facts. Perhaps then the ultimate sensory experiment involving the Freemartin on the farm has been an unintentional embrace of the animal's misuse of time. I think of @guy number 2 and his description of his Freemartin Rosie in one of the online farm forums. Supplemented with testosterone and juiced up like a male full of machismo, sure enough, she went from mildly dainty to distractive in a matter of days, he says. She pawed the ground like a bull. She growled 2 octaves lower like a bull. She threw her head around and acted tough like a bull. And yet, despite attempts to reattune Rosie's body, @guy number 2 admits that she became difficult to herd with an ATV like a bull, but she did not develop an attraction for cows like a bull. No interest in them whatsoever. The Farm Forum is an archive to a morphology as it has occurred within language, around the Freemartin sex and sexuality. I notice a preoccupation with understanding animal passion. And like the different kinds of scientists who have engaged in Freemartin research, these online farmers invest in Freemartin desire as a significant and productive industrial force, thus making the barren Freemartin once again a problem worth addressing. In describing animal passion, many members of the farm forums favor a similar tendency towards adjectives reserved for human sexual behaviors, as others did before them. Bulls are fiends, sex is coital, and heat is the climactic outpouring of the female's desire. At guy number 2 was forced in the first place to use Rosie as his Gomer. As he puts it, his bull was not a player. His bull, he says, wanted a deep, meaningful, monogamous, long-term relationship. We see here how long-standing human gendered stereotypes are grafted onto the behavior of non-human animals. Animals are enlisted to deliver truths about gender, like hippos served watermelons at the zoo that burst in their faces with baby boy blue. This time, at guy number 2 grafts human ideas about gender in the other direction onto the behavior of non-human animals. I'm sorry, but a lesbian cow, concludes at guy number 2. That's a funny story. I don't care who you are. As we see, ideas about human gendering flow multidirectionally between human and animal, like hormones across the fetal membrane. We've seen that knowing the Freemartin has involved using the senses as a means of interpretation. But extracting value from it has also involved transforming the animal itself into an interpretive tool. In both these cases, the Freemartin is an interface between human and non-human animal experience, often enlisted as a means of finding coherency out of confusion. And yet the Freemartin is an anomaly. The Freemartin is allowed to misuse time. And this non-productive use of time has arguably made the animal most prolific. The Freemartin's taste, tone, and texture are also indefinite. And what seems most notable is not the many achievements and significant discoveries that were made in trying to fix or attend to the animal's unproductive body, but rather the emphasis that the Freemartin puts on the productive potential of its own instability. The Freemartin's incoherency forces for an understanding of sex as continuous rather than divisible into distinct categories. And with that, the production of sex is not only genetic, but also an environmental process. Attempts to fix or grasp this liminality have been geared towards controlling dominant ideas about gender and sexuality and reinforcing normative values at human, bodily, and social scales. But on the whole, engaging the senses, it seems, has been a means of not letting the Freemartin slip through the hands of the various actors that try to define it. Unlike the scientist Frank Rocheret-Lilly, paying attention to the Freemartin here has gone beyond recording its dysfunctionality. Examining the Freemartin here has presented a stretching out of time beyond its instrumental uses and the reinsertion of pleasure on the farm.